Hi guys, welcome to chapter 2, cell structure and function. This particular chapter has four parts. The first one looks at cell types, the second one looks at cell size and compartmentalization, the third one looks at organelle structure and function, and then the last one looks specifically at the plasma membrane. We're going to start with 2.1 cell types. Let's jump into it. Okay, so basically cells are the basic structural unit of all living things. And the cell theory states that all organisms are composed of cells. Cells come from pre-existing cells, which is what we call biogenesis. And the cell is the smallest living organizational unit, right? When I'm using the term organism, I'm referring to any sort of living thing, whether it be a plant cell, an animal cell, or bacterial cell, and so forth. It was previously thought that cells could spontaneously generate from the environment, but in 1859, Louis Pasteur disproved this theory by boiling beef broth in the swan neck flasks and proving it otherwise, right? How the experiment was designed was pretty simple. Basically, he heated this kind of beef broth in this swan neck flask. The little swan neck is here. And if you just leave it, bacteria can't get in, proving that the design of the swan neck and the moisture levels that are in there prevents bacteria from getting in. However, if you removed it, the bacteria did get in and therefore managed to move into that particular spot and boil the broth. And if you also applied the heat and then tilted the swan neck just to expose it past the neck, that kind of curve in the neck, and then you it back upright, the bacteria still managed to get in and populate the broth anyway, right? So that gives you a bit of an idea as to how he managed to figure it out. The term pasteurization was also named after Pasteur. All cells have common features, right? It doesn't matter whether it's a plant cell, an animal cell, or bacteria cells, they all have these four things. They all have a plasma membrane, which separates the cell from its environment, much like a skin of an organism does. It has a cytoplasm. Now, the word cytoplasm is the collective term for the insides of the cell, right? Which includes things like the organelles, which are specialized structures in the cell, or the cytosol, which is this kind of gel-like substance comprised mainly of water. The term cyto is referring to cell, right? So it's a cell plasm or cell goop, if you would. All cells have DNA, deoxyribonucleic acid, which is the genetic information of all living organisms, and all cells have ribosomes, which are specialized organisms responsible for the synthesis of proteins. Very important for living things. From there, you notice fundamental differences between different types of cells. For example, plant cells and animal cells are different in the sense that animal cells don't have a chloroplast, and they also don't have a cell wall, which plant cells do have, making them different from animal cells. However, bacterial cells don't have mitochondria, which plant cells and animal cells do have. There are fundamentally two different types of cells. You've got your prokaryotic cells and you've got your eukaryotic cells. Prokaryote refers to before the nut or the kernel, karyon being nut or kernel, or referring to specifically the nucleus. So a prokaryotic cell lacks a nucleus. It is generally small in size and simple in cell structure. It includes things like bacteria and archaea. They're unicellular and are single-celled organisms, and they also lack particular organelles like the mitochondria. The DNA is in a particular region called the nucleoid and is made up of these rings called plasmids. Here are a few examples of prokaryotic cells. You can see they just kind of look very ordinary, very simple. Eukaryotic cells, on the other hand, are a lot more sophisticated. The word eukaryote meaning true nut or kernel. They are much larger in size and more complex. They have the nucleus. It includes all the other kind of things that are out there, animals, plants, fungi, and protists, and they contain membrane-bound organelles like the mitochondria and the chloroplast. They can be both unicellular or multicellular, and the DNA are in the shape of chromosomes encased inside a particular nucleus, right? Which we will cover in the next chapter. And here is the plant cell, and these little kind of green beads are the chloroplast, and this is a protist. And you can see there the organelles that are found in there as well, including this particular cool looking thing called an oral groove, which it uses to eat food. Here is a diagram showing you the difference in complexities between the two. You can see the prokaryotic cells are very simple. Your bacteria, there's your nucleoid region. Your cytoplasm is pretty empty, and then your cell is a lot smaller. Whereas your eukaryotic cell, you see a lot of happenings going on there. The nucleus with the nucleolus, and then also the endoplasmic reticulum and mitochondria and so forth and so forth. Okay. Let's take a look at some prokaryotes that we are familiar with, bacteria being the primary example of a very typical prokaryotic cell. Bacteria are unicellular and microscopic. They are prokaryotic, and they were the first living organisms to have appeared on the Earth about 3.5 billion years ago, and they're also the most numerous type of organisms today. Collectively speaking, there are more bacteria in weight than there is every other living thing in the world put together. Right, which is pretty crazy because you can't even see bacteria. Bacteria play a really important role in the ecosystem. They break down substances such as plant and animal waste and recycle material, as well as become a food source for other organisms as well. Here is a diagram of how bacteria divide. It's a process called binary fission, which we will look at later. But you can see they kind of elongate and they break up from 1 to 2, 2 to 4, 4 to 8. And within a day, you can have upwards of 10,000. Okay. Here's another little animation of a bacterial cell being eaten actually by one of your white blood cells. So there's your little bacteria getting glooped into a white blood cell and getting eaten into it. And there's another one there kind of getting around, stuck into it. And there's your white blood cell kind of consuming the bacteria that's there. Here's the diagram showing you the amount of bacteria that are around. You can see there are way more bacteria than there are plants and animals in the world today. There are two types of bacteria. You have what we call gram-positive bacteria and gram-negative bacteria, and it all comes back to this particular layer in their cell wall, which is called the murine, which is a type of sugar protein. Basically, gram-positive bacteria have a thicker layer of murine, and this thicker layer allows them to absorb a particular material called crystal violet, which scientists use to make the bacteria appear on their microscopes as purple. So a gram-positive bacteria appears as purple, and it includes examples like Staphylococcus, which is what the staph infection bacteria is, and a few other things as well. So for example, here's your gram-positive, so staph is here. Anthrax, which is also another type of bacteria, is a gram-positive bacteria. Pneumonia is another one here, as well as a few other ones like diphtheria and so forth and so forth. Okay. Gram-negative bacteria, on the other hand, have a thinner layer of murine in their cell wall, which as a result absorbs less of the crystal violet stains, and instead of appearing purple, which is the darker color, they appear pink, which is the brighter color and less concentrated. Right. So examples of gram-negative bacteria include things like cyanobacteria, meningitis, and a few other things as well, like E. coli and so forth. Right. Here is the diagram showing you the difference between that particular layer. So you can see there, apart from the outer membrane, your gram-positive ones, they have a lot thicker of this kind of sugar protein here. That's the murine, and then the gram-negative ones are a lot thinner. Okay. Aside from the bacteria, another group of organisms that fall under the prokaryotic branch is the Archaea. Archaea are also simple single-celled organisms that are prokaryotic, and they're usually classed as what we call extremophiles. Extremophiles are able to handle the most extreme environmental limits on our planet. Extreme and phile meaning love, so they love kind of extreme things like high and low temperatures even in the atmosphere, in acidic and alkaline environments, salty environments, dry environments, areas when they're light, and also things like deep underground. Now this is due to their unique plasma membrane that remains fluid and permeable over a wide array of temperatures, allowing them to survive really well. Okay, so here's a few examples of Archaea. These ones are drawn from hydrothermal vents at the bottom of the sea floor. Pretty remarkable, and these are kind of these hydrothermal vents are shooting out like ash and sulfur and things like that, and the bacteria feed off, and the Archaea feed off these particular locations. It's pretty cool because it actually forms the basis of an underwater food chain where that gets absolutely no sunlight, and and then there are organisms like worms and things like that that feed off those Archaea, and it starts a brand new food cycle. Well, that's it. I hope you guys enjoyed this particular video, and I'll see you guys in the next one.